Hello, my name is Nathan VanDusen. I'm an assistant professor at Indiana University, and today I'm going to be talking about some of our work where we're seeking to decipher a CRISPR-Cas9-mediated precise genome editing mechanism within the heart. So in this project, we're using CRISPR-Cas9 to create DNA double-strand breaks, and then typically these breaks get repaired in one of two ways. The most common way is non-homologous end joining. In this method, the cell basically just rejoins the two ends of the broken DNA strand, and in the process, some small insertions or deletions typically get created. So this is a very error-prone repair pathway. And then alternatively, and what we're trying to engineer improvements in this pathway, or improvements in efficiency, the alternative is homology-directed repair. And in this pathway, you supply a donor template, or the homologous chromosome gets used as a template, and the break uses that template to get repaired in a very precise way. And so in this fashion, you can engineer very precise changes to the genetic sequence. Okay, so previously in my research, I had shown that we can use adeno-associated virus to carry a DNA donor template to trigger homology-directed repair within mouse cardiomyocytes. And so to briefly recap some of these experiments, we had used this AAV vector carrying homology arms targeting the MYL2 gene locus. And after creating a DNA double-strand break with a CRISPR and a guider in it, we can get insertion of a self-cleaving 2A peptide along with a red fluorescent protein. And so when we target the MYL2 locus in this way, you can see expression of our red fluorescent protein specifically within the ventricle, not within the atria. And this reflects the native expression profile of the MYL2 gene. We also went on to test this at different loci, one of them being MYL7. This is a gene that's specifically expressed within the atria. And so again, we could see efficient insertion of RFP at this loci, and we get strong expression within the atria, but not the ventricle. So this was in neonatal cardiomyocytes, but we also went on to show that it works at similar efficiencies even within adult cardiomyocytes. And this was really surprising. It was previously thought that as postnatal cardiomyocytes withdraw from the cell cycle, that a lot of the molecular machinery that's required for homology-directed repair would not be active and not be present. And so it was thought that adult cardiomyocytes were not compatible with homology-directed repair. We showed actually that's not the case, at least within this AAV-mediated mechanism. And so really that suggests that there's some sort of non-canonical HDR mechanism happening here that we don't understand very well. We also went on to test this approach at many different loci, and one of our key observations was that the efficiency of the approach was directly proportional to how highly expressed the gene was. And there are some different ideas about why this might be. Probably the most likely is that the chromatin may be more open and accessible at more highly expressed genes. But again, this is something else about the mechanism that we don't really understand very well. Okay, so there are many different applications of this technique. One is disease modeling. We've since gone on to show that we can create specific mutations that have been associated with human disease within mouse cardiomyocytes. Here, shown on the left, we have a phospholamban arginine 14 deletion mutation fused to M scarlet red fluorescent protein. And so you can see the cellular localization of the mutant protein. And then using single cell phenotyping assays, we can detect phenotypes that are associated with this particular mutant. In this case, we see calcium alternans, which is where we see this variation in peak amplitude from beat to beat within these mutant cardiomyocytes. And so this is a phenotype that's highly associated with arrhythmia susceptibility, which is something humans with this mutation suffer from. Okay, so beyond disease modeling, there are other additional applications like functional genomics that I'm going to talk about shortly. And then the other application that I'm really interested in is therapeutic applications. And so what we would like is to be able to precisely deliver therapeutic transgenes to safe harbor loci using this approach. But before we can use it therapeutically, we really need to better understand the mechanism. Like I previously mentioned, there's quite a bit of evidence suggesting this is a non-canonical HDR mechanism. And then if we can understand the mechanism better, hopefully we can also engineer higher efficiencies to make any therapeutic approach more effective. Okay, so to understand the mechanism of this genome editing approach, we set out to develop a functional genomics-based high-throughput forward genetic screen for regulators of the process. And this approach, again, uses CRISPR-Cas9 to create targeted DNA double-strand breaks. These breaks will primarily be repaired by non-homologous end joining, which creates small loss-of-function mutations within the target genes. And so what we did is we designed a library of guide RNAs targeting all known mouse DNA repair factors, packaged that library within an adeno-associated virus, and then delivered it to newborn mice expressing Cas9. And so this is a depiction of the result. The idea is that we're creating different mutations in each cell within the mouse heart. Some of those mutations will affect HDR efficiency, some will not. Okay, and so this is the full genetic screening strategy. This is a novel functional genomics-based assay. And again, just to recap, we're delivering a library of guide RNAs to newborn mice expressing Cas9. This creates the mutations. We're then waiting 10 days to allow for depletion of the protein, and then we're injecting a second virus, and this virus carries a HDR donor template for insertion of the transcriptional activator TTA into the MYL2 locus. There are basically two outcomes. If there's non-homologous end joining, there will be no insertion of TTA, and so the transcriptional activator won't be able to activate a TRE responsive reporter gene within our first AAV vector. So this reporter gene expresses a red fluorescent protein, and then it has the library of guide RNAs embedded within its 3' UTR. So if there's non-homologous end joining, there's no activation of the reporter gene on the first vector. However, if there's homology-directed repair, then we get insertion of TTA into the MYL2 locus, it gets expressed, and then it activates the reporter gene in such a way that we get production of these reporter gene transcripts that have a non-functional copy of the guide RNA, basically barcoded within the 3' UTR of the reporter gene. And then we can very easily measure the frequency of each guide RNA within the pool of reporter gene transcripts by amplicon sequencing. So the whole idea here is that guide RNAs targeting genes that are necessary for HDR will be depleted within this pool of reporter gene transcripts. And then guide RNAs targeting genes that antagonize HDR, mutation of those genes will actually result in higher rates of HDR. So those guide RNAs will be enriched in the pool. Okay, so first we set out to validate our novel system. First we injected the vector carrying the guide RNA library and the red fluorescent protein reporter gene. And you can see when we inject this vector by itself, there's no red fluorescent protein expression, indicating that the reporter gene is not leaky. And then if we inject the guide RNAs on the reporter gene in combination with a second virus that just expresses TTA, we get indiscriminate activation of the red fluorescent protein reporter gene, as you can see here. And then finally, for the actual experiment, we delivered our library of guide RNAs and the reporter gene at birth, 10 days later, we're injecting our HDR donor virus and getting precise insertion of TTA within the MYL2 locus. And so that results in a strong expression or strong activation of our reporter gene, specifically in the ventricle. We don't see expression within the atria. So this middle panel here, these are our control group samples. They have indiscriminate activation of the reporter gene. These are our screen knockout samples. They only have activation of the reporter gene if homology-directed repair has successfully taken place. Okay, so we first looked at the expression of individual guide RNAs within our library, and particularly the control guide RNAs. So we had two negative control genes, TTA and Cas9. If either of those genes is mutated, there should be no HDR or no activation of the reporter gene. So those guide RNAs should be depleted. And you can see we have 8 guide RNAs for each gene. So there's some level of redundancy built into the screen. And we were able to observe that the vast majority of guide RNAs targeting TTA and Cas9 were depleted within our screen. Next, we looked at Fanconi anemia factor A. So FANC-A, this gene had been previously associated with homology-directed repair, or specifically AAV-mediated homology-directed repair. So this is one of the very few genes that was previously known to play a role in the process. And our screen results supported those previous data. We saw that most of the guide RNAs targeting this gene were depleted, at least modestly. And then we had 20 scrambled control guide RNAs, so these are not targeting the mouse genome. And you can see that on average, there was no enrichment or depletion of those guide RNAs. So the control guides seem to do what we were expecting them to do. So we moved on to looking at all of the other guide RNAs in the library. And what you can see, I've just highlighted some of the interesting things in color. The red dots are genes or guide RNAs associated with homologous recombination. These tended to be strongly depleted, which is consistent with the idea that they're targeting genes that are necessary for this process. And then we saw some guide RNAs associated with non-homologous end joining. These guides were highly enriched, which is consistent with the idea that non-homologous end joining is a competing pathway. So if we disable that pathway, we'll get increase in HDR efficiency. And then surprisingly, we saw some factors associated with monoubiquitination. Those are shown in blue. And then we saw several factors associated with the Fanconi anemia pathway. And as I previously mentioned, there has been some previous evidence that the Fanconi anemia pathway is important for this AAV-mediated HDR process. The Fanconi anemia complex is quite large. There are lots of different factors, and we observed guide RNAs that were both enriched and depleted, targeting various factors. So the role of Fanconi anemia pathway in this mechanism is unclear and probably fairly complicated. Okay, so those were individual guide RNAs. I had mentioned there are 8 guide RNAs for each gene. So we used a statistical analysis package called MAGIC, which consolidates the 8 individual guide RNAs per gene into a single gene level score. And when we do that, we got 13 genes that were statistically significantly depleted, and then 11 genes that were significantly enriched. For our depleted genes, there were UBE2A and UBE2B. These are genes that are associated with protein ubiquitination that were among our top hits. And then a range of factors in the enriched gene set, including Fanconi anemia factor C. When we do gene ontology analysis, so in this case, gene set enrichment analysis of our screen results, we saw that guides associated with homologous recombination tended to be depleted. Factors associated with ubiquitin-mediated proteolysis and mismatch repair were also more modestly depleted. And then we saw some enrichment of factors associated with non-homologous end joining, base excision repair, and nucleotide excision repair. So this was in line with what we had previously observed just by looking at our screen hits individually. And so it was quite surprising that these ubiquitin-associated factors had come out as such strong hits in our screen. And particularly, we were interested in these factors UBE2A and UBE2B. And we would like to basically look at the possible roles they may be playing in this mechanism and identify where their essential function is really coming from. So one possibility is that UBE2A and UBE2B interact with RNF20 and RNF40. This is a well-known interaction. Together, they monoubiquitinate histone 2B, and this is an activating chromatin modification. And so it's been previously thought that possibly this modification could enhance HDR through basically relaxing the chromatin and opening it up, making it more accessible. So that's one possibility. It's not entirely consistent with our screen results in that we saw strong depletion of UBE2A and UBE2B, but we did not see depletion of RNF20 and RNF40. But nonetheless, it's one possibility. Another possibility, UBE2A and UBE2B are known to interact with the DNA repair factor RAD18 to monoubiquitinate pCNA. pCNA is a very critical protein involved in various aspects of DNA repair as well as DNA replication. So it could be regulating the function of pCNA. And then a third possibility, so I mentioned the Fanconi anemia pathway was previously known or previously shown to be involved in this AAV-mediated homology-directed repair. And the key regulatory step for the Fanconi anemia pathway is ubiquitination of FANCD2 or FANCI. And so it has not been previously shown that UBE2A or UBE2B can ubiquitinate these factors, but there could be either a novel interaction happening there, or UBE2A and UBE2B could just more broadly be affecting ubiquitin dynamics in a way that impacts the Fanconi anemia pathway. And then the last possibility is that UBE2A and UBE2B are interacting with some other unknown factors. All right, so for our future plans, first we need to validate our screen results. To do this, we'll individually knock out some of our top screen hits and then see what the impact of that gene knockout is on the efficiency of homology-directed repair. And then once we validate our screen hits, we plan on testing possible roles for UBE2A and B, as I mentioned in the previous slide. One way we can do this is to knock out RNF20 and 40, and then measure the impact on AAV HDR. We could also knock out UBE2A and B using our CRISPR somatic mutagenesis system, and then see what impact that has on the ubiquitination of pCNA. And very similarly, we can use the same strategy to see what the impact is on ubiquitination of these Fanconi anemia pathway factors. And then if it seems like UBE2A and B aren't acting through either of those three mechanisms, we could potentially do some proteomics to try and identify novel UBE2A and B interacting factors within cardiomyocytes. And then more long-term, we would really like to discover some strategies for enhancing AAV HDR efficiency. So to do that, one mechanism or one approach now that we've validated our functional genomics assay is we can expand our genetic screen to all genes known to encode druggable factors rather than just screening the DNA repair factors. And so the hope here is that we can identify some factors that antagonize AAV HDR, and then by a druggable perturbation, we can block the action of those factors and get an increase in AAV HDR efficiency, which again would be very useful for translating this approach to therapeutic applications. Okay, so with that, I just want to acknowledge everyone in my lab who's done a lot of hard work on this project. Sanchita Day is the postdoctoral fellow who's done most of the work for this project, so I really want to recognize her efforts. And then we've also been funded by a variety of sources from the National Institute of Health. So with that, just thank you for your time, and I'd be happy to take any questions.